Swayam Prabha. Digital India. Educated India.
Hello, my dear students. Welcome to Swayam Prabha, sponsored by MHRD, New Delhi. Course title Medicinal Chemistry. Lecture title Principle of Drug Design. Faculty name Dhamak Vikrant Murlidhar. Module number 6, lecture number 9. So today we will be looking principles of drug design. So the contents of the today's lectures are structure-activity relationship. So we can say structure-activity relationship, it is a heart of medicinal chemistry. Shortly it is said to be SAR. Factors considered for SAR. After that we will be looking for lead compound, its utility and properties. Methods of identification of lead compound. So the first point we will be discussing from this session, that is structure-activity relationship. So name itself indicate that activity of any compound or any molecule does depend on the structure, and the groups responsible for its activity in the structure, so that is to be studied by a relationship. So which groups are responsible for what activity, or which groups are needed for an activity, so such kind of a relationship we have to study in this session. Structure-activity relationship is the relationship between chemical structure of molecule and its biological activity. As discussed that chemical structure, as the chemical structure changes, the biological activity will be either increased or decreased. So it depends on an chemical structure and what group does it require for an biological activity. So such type of a relationship, it is said to be an structure-activity relationship, or shortly said to be an SAR. This idea was first presented by Brown and Farsner in 1865. Analysis of SAR enables determination of chemical group responsible for evoking target biological effect in the organism. Say suppose a compound, it is having some functional group in it, or some groups are responsible for an activity. So SAR studies that makes and researcher or medicinal chemist to know that which groups are responsible for what type of an activity, or what chemical group from that molecule is attaching to a target or fitting with a target, and due to which biological effect has been shown in the organism. So once we know what groups are responsible for an activity in the biological target, then medicinal chemist can make a modification of the effect or the potency of an bioactive compound by changing its chemical structure. Once a medicinal chemist he knows that what type of groups are responsible for an activity. So similar or medicinal chemist can do an modification by to increase an potency or bioactivity of a compound. Like if you take an example of an fluoroquinolons. So fluoroquinolons were only firstly an quinolas, but by introduction of an fluorine group in fluoroquinoline, like example of an ciprofloxacin or norfloxacin, when fluorine group it was been introduced, so it increased in lipophilicity, and due to which the activity of an quinolones was increased, and due to that ciprofloxacin drugs like ciprofloxacin, norfloxacin, sparfloxacin, a series of floxacin it was been developed. So groups which are responsible for an activity, so it has been studied for by an SAR. We will be discussing that how SAR works. Minor modification in a drug molecule, and this minor modification changes a major change in pharmacological activity, as discussed that fluoroquinolons. So quinolones were only firstly a drug class, but later on by introducing an fluorine group at in the ring of an quinolone, so there was major change in pharmacological activity. So similar SAR works. Medicinal chemists use the technique of chemical synthesis to insert new chemical groups into a biomedical compound and test the modification of their biological effects. So this is one of an advantage of an SAR, that once a medicinal chemist knows what groups are responsible for what type of an activity, whether it increases lipophilicity, whether it decreases hydrophilicity, or whether penetration in the brain it increases, or whether interaction with a receptor it increases by introducing some groups or making a minor modification in a bioactive compound. So medicinal chemist has a knowledge of a chemical synthesis, and due to which the medicinal chemist can insert a new groups into the lead compound, and once the groups are being introduced responsible for an activity, so there will be modification in biological effects also. So we'll look towards an example of an SAR, so how it works. So one of a drug, that is heroin, is weak mu agonist, mu receptor agonist. 3,6-diacetyl derivative of morphine. If we look towards a structure of an morphine, so this is a structure of an morphine, and instead of this hydroxyl group over here, so it has been substituted by diacetyl derivative or diacetyl group, and this 3 position and 6 position makes it more lipophilic by introducing this diacetyl group at position 3 and position 6. So here these groups are responsible for an ionic bonding, but by introducing such type of an acetyl group, so it increases an lipophilicity of a molecule, and due to which lipophilicity of a molecule increases, so the interaction will be similar such as morphine it is giving, but by introducing such type of groups into a molecule, so it will increase an lipophilicity of a molecule, and due to which it shows an activity. So heroin, it is more potent than morphine, and there are 2 factors which make it more potent. So one of the factor, that is it increases lipid, and which enhances its penetration into a brain. So these acetyl groups are responsible for increasing in lipophilicity, but while this hydroxyl group, they are responsible for an ionic bonding, and due to increase in lipophilicity, so molecule becomes more lipophilic and it can easily cross blood-brain barrier. And also, apart from this lipophilicity, metabolite of this 6-acetyl morphine is more active than morphine. So metabolite is also active, and with that it increases the lipophilicity. So these are the two advantages by introducing an acetyl group. So now we will look towards a point, what is need of SAR, why medicinal chemist requires an SAR. Study of structure activity relationship is mainly done on lead molecule. So lead molecule we will come across in this session what is actually a lead molecule. And so the lead molecule is such type of a compound. So once medicinal chemist can discover it through some process, and once lead molecule it has been developed, from that pharmacophore it has been developed. And the structure activity relationships are mainly done on lead molecule. So in this session later on we will see what is actually a lead molecule. So need of SAR, secondly, it is that to develop a new drug that has increased activity. So groups responsible for an activity, once a medicinal chemist knows it by making a minor modification. So medicinal chemist can introduce such a group which increases the activity of a molecule. To know the changes in pharmacological properties by performing minor changes in a drug molecule. Also, we will come across in this session that by inserting or by removing some groups, there is change in biological activity also. So the molecules, one molecule can show or similar kind of a lead molecule can show different effects by changing just only and some groups, and it will show a different activity. Also, along with that, pharmacological properties can be changed by making and minor changes in a drug molecule. So like if you take an example of a stereochemical drug, suppose an enantiomers. So there are two enantiomers which are responsible for an activity, but like in some case of an, now we are coming across that earlier it was just an cetirizine in the market. Cetirizine, everyone is familiar with that drug, that cetirizine, and it is one of an antihistaminic drug. Nowadays we will see in the market that it is been sold with an leocetirizine. So from that it can be understood that just making a minor changes. So minor change, it means not only by introducing group but also by use of an stereochemistry also. So it will perform and a different biological activity, or it will show little bit change in pharmacological activity. To determine some different activity of from an existing drug, like it, there was an example of an sulfonamide in Second World War. So sulfonamide, it was used as an antibacterial agent, but later on it was been found that soldiers who were using sulfonamide as an antibacterial drug, so sugar levels was been decreased, and this sugar level was been decreased because sulfonamide also has an anti-diabetic property. Later on it was been introduced in the market as an drug of an anti-diabetic properties, and also it has been marketed as a drug of an antibacterial property. So making a minor modification with the sulfonamide structure, basic lead molecule of an sulfonamide, so there was, so it showed an potent anti-diabetic properties also, and along with that it also showed an antibacterial activities also. So earlier it was used only an antibacterial, but later on it showed an anti-diabetic activities also. And also to minimize the side effect, so we will be coming across one of a drug, that is thalidomide. So how the side effects has been minimized by using a study of an SAR. So we will coming across with that also. Now we will be coming towards a point, that is SAR studies. So there are 1,000 molecules, 100 molecules, and from this chemical compounds, the screening it has been done, and randomly they are been screened. After that, a lead molecule, it has been identified. So lead molecule is a molecule which shows an biological activity. And after that, by pruning process, so pruning here, it means that by cutting or by inserting some groups into that lead molecule, it develops an pharmacophore, and that pharmacophore, it is nothing but a drug. And pharmacophore, we can say it is such a moiety that what kind of groups are responsible for an activity. So pharmacophore is a study of such a kind, and it is done with the help of a pruning process. So now we will be looking what are the factors considered for SAR, structure-activity relationship. Size and shape of carbon skeleton. Secondly, nature and degree of substitution. After that, stereochemistry of molecule. So these are the factors considered for SAR. So we will be looking one by one. Changing size and shape. Now we will discuss point changing size and shape of a molecule. So what are the strategies in changing size and shape of a molecule? So first strategy, that is number of methylene groups in chain and rings. Increasing or decreasing degree of unsaturation. So that is either removing a double bond or inserting in double bonds. Introducing or removing a ring system. So we'll look towards an first strategy, that is number of methylene groups in chain and rings. So we'll see with an example. See, here it is an example of an variation in methylene groups. So it is a structure of an polyphenols or resorcinol, and it is showing an antibacterial activity. So this graph, it is indicating antibacterial activity versus number of N. So versus number of N, it is meaning that number of N methyl groups. So when methyl groups are 1 and 2, the activity is similar. As it becomes, as the group becomes 3, so activity increases, and as group becomes 4, again activity increases, and maximum activity it is shown when methylene groups are 6 in number. And as methylene group increases, the chain increases by an 7, so the activity decreases, and as it becomes 8 group, so again the activity decreases. So the meaning of this thing is that specific number of groups are responsible, specific numbers are responsible for an activity. So it does not mean that the graph will be always linear with increasing number of methylene groups. So it will decrease after certain numbers, and here it can be seen that as methylene it becomes 6, methylene groups become 6, so it shows a maximum activity, and as it becomes 7, so there is increase, so there is decrease in activity, and as also it becomes 5, so again there is decrease in activity. So maximum 6 groups or 6 methylene groups are responsible for an highest activity. So increasing or decreasing beyond the 6 or below the 6, it decreases the activity. So it can be understood from this graph that methylene groups, or we can say straight chain, it is responsible for an activity but up to unlimited numbers only. So that is why structure activity relationships are important. So here the structure activity relationship shows that when there is number of groups or methylene groups, when it is up to 6, then only it shows an highest activity. Beyond it or below it reduces the activity. So next point, that is increasing or decreasing the degree of unsaturation. So degree of unsaturation, it means that by increasing or decreasing the double bonds. So here you can see in the example, so these are steroidal structures having 4 rings, and so in that Cortisone, it is not having a double bond over here, but while prednisone, it is having a double bond over here. So there are two activities of the compounds. So one is glucocorticoid activity and second one is an mineralocorticoid activity. Glucocorticoid activity of prednisone increases than cortisone due to double bond. Cortisone and prednisone. So the prednisone shows increased activity of an glucocorticoid due to presence of a double bond, while mineralocorticoid activity of both drugs are similar. So there is no change in mineralocorticoid activity, only there is change in glucocorticoid activity. And as the double bond it has been introduced, so it becomes more rigid. The rings become more rigid than just not having a double bond over here. So ring becomes more unsaturated and it becomes more rigid by introducing a double bond. So here it can be understood that by increasing or decreasing of an unsaturation, the activity changes. So minor change just only of a double bond and the activity of a molecule changes. Rather all molecule it is similar. Next part, that is introducing or removing a ring system. So we will be coming across an example of an penicillin. Benzyl penicillin is not resistant to beta-lactamase, but by introducing a phenyl ring in a molecule, so it forms a new structure, that is dipenicillin, becomes resistant to beta-lactamase. So these are the structures of an benzyl penicillin, and second, that is dipenicillin. So introducing an phenyl ring over here, right? In case of benzyl penicillin, here you can see that there is no phenyl ring, but in case of an dipenicillin, there is an phenyl ring. By introducing this phenyl ring, so there is resistant to an beta-lactamase. So you might have come across that whenever there is when we take a marketed formulation of an penicillin, so always it happens that it is having one more API, that is active ingredient, and that API it is nothing but an clavinic acid. And so the introduction of an clavinic acid in that formulation, it is for and this beta-lactamase, because once a molecule it comes in a contact in the biomembrane, so beta-lactamase, it is an enzyme which deactivates this penicillin, but clavinic acid it prevents from deactivation of this beta-lactamase of an penicillin. But by making a minor modification, that is introducing an phenyl ring, so it becomes resistant to an beta-lactamase. So here also it can be understood that by making a minor change or of removing a ring or introducing a ring, so there is difference in activity, that this structure it is not having a resistance to an beta-lactamase, but this dipenicillin has a resistance to an beta-lactamase. Now we'll move towards the next point, that is stereochemistry of a molecule. Different activity are shown by a molecule with a change in stereochemistry. So here it is an example of an enantiomers, racemic mixtures, RNS, which are the mirror images of each other, but they are non-superimposable, and having a similar structure, only it rotates and plane polar light towards and right, and this rotates and plane polar light towards and left. So it is S enantiomer and this one is an R enantiomer, but there is difference in activity. R-ketamine, it is responsible for hallucinogenic activity, or it can be said antipsychotic activity, and S-ketamine, they are responsible for anesthetic activity. Both these drugs act on CNS, but there is difference in activity, that one is antipsychotic and second one is an anesthetic. So different stereoisomers have a different activity. So from this it can be understood that these two isomers are having different activities. Now the second part will be looking in stereochemistry of a molecule, that is no activity of one isomer. This LS alpha methyl dopa, it is an antihypertensive and mirror image of this molecule non-superimposable, that is an enantiomer. R isomer, it is inactive, but S isomer, it is active. And this is why sometimes we see in the market or we see in the formulation that particular isomer has been only been utilized, and this is the reason why medicinal chemists from studies, they come to understand that which type of an isomer is responsible for and what type of an activity, and the compounds are being synthesized in such a way only that it shows maximum biological activity. So here you can see that there is change in configuration of this molecule due to chiral center, and which shows a different activity, or R is inactive while S is active. And this is because the groups are responsible, specific groups are responsible of an pharmacophore for attaching or for interacting with an receptors, and due to interaction with an receptors, so it will show an activity, biological activity. So in earlier session also we have seen that how molecule attached to an receptor or interacts with an receptor, and due to which molecule is responsible for an activity. So from this example, it has been understood that not all the isomers are responsible for activity, but in case of some isomers are only responsible for an activity. Now we'll see third point in stereochemistry of molecule, that is stereoisomers showing side effect. So this is a structure of an thalidomide. It acts on CNS, while the mirror image of is, that is S-thalidomide, it is teratogenic. So from this it can be understood that the stereoisomers, all the stereoisomers are of not of all medical uses, but like in case of this one, it is teratogenic, and while R isomer, it is showing an activity. So teratogenic, it means that in the fetal or newborn baby, there are some defects in the limbs, that is said to be teratogenic. Now we'll come towards a point, that is nature and degree of substitution. Third point. Now from this table, one can understand how to increase a lipophilic character and how to decrease an hydrophilic character. So methyl group, when methyl group it has been introduced, so it increases an lipophilic character, but it decreases the water solubility. And so the meaning of this thing is that, so both the things, it will not happen at the same time, means by introducing certain groups, so it will not happen that lipophilic character will also increase and hydrophilic character will also increase. So there will be increase and decrease, either one will increase and second one will be decreased. So lipophilic character has been increased by introducing an methyl group, and decreased the water solubility, means hydrophilicity has been decreased. Fluorine and chlorine. So earlier in this session also we have discussed an example of an fluoroquinolons. By fluoro itself indicate fluorine, the molecule it is having an fluorine, and due to increase in lipophilic character of an quinolons, so it formed an fluoroquinolone, and which showed an increase in antibacterial activity. So ciprofloxacin and norfloxacin are such a drug having an fluorine at 6th position, and due to which the activity was been increased. But by increasing lipophilic character, there is decrease in water solubility. After that, hydroxy group decrease lipophilic character. So by decreasing an lipophilic character, it increases an water solubility. After that, amino carboxylic acid and sulfonic group, it also decrease lipophilic character, because this all four, hydroxy, amino carboxylic acid and sulfonic acid, so these groups are responsible for an ionic interaction. So these are responsible for interaction with an amino acids, but due to dissociation with an body fluids, so it increases only an hydrophilic character, that it increases water solubility, but it decreases in lipophilic characters. To be an molecule of an lipophilic character, so non-polar groups should be introduced into an molecule, so only it will increase in lipophilic characters. So here these are non-polar groups, methyl and chlorine or another halogens also, which are responsible for lipophilic character in the molecule. But remaining these four, these are increasing hydrophilicity and decreasing in lipophilic character. So we'll see one of an example of nature and degree of an substitution. So it is an basic analog or basic moiety, that is 4-amino antipyrine, and this 4-amino antipyrine with a substitution shows and different activities. So we'll see one by one. This 4-amino antipyrine analog, a basic moiety, it shows an antioxidant properties when chromate has been introduced. After that, by substituting it by an phenyl rings, it shows antimicrobial activity. Again, by substituting this, it shows analgesic activity. Anti-inflammatory activity. After that, cytotoxic analog. After that, anti-enthelmintic activity. After that, antitubercular property. After that, antiviral activity. So from this, it can be understood that it serves as a lead moiety, or we can say an pharmacophore, and by introducing certain groups and at certain positions on this 4-amino antipyrine, it shows different types of an 8 activities, and these different 8 activities are responsible because are been shown by a molecule, because there is such type of an introduction of an moieties which are responsible for an interaction with an receptors at that particular site, and due to which it shows and this different activities. So from this, it can be understood that by substitution of an groups, or by substitution of an, or by introducing an different moieties in a basic molecule or in a lead molecule, so it shows in different activities. After that, we will be studying what is lead compound. Lead compound is generally defined a new chemical entity that could potentially be developed into a new drug by optimizing its beneficial effects and minimizing its side effects. Lead compounds are typically used as starting points in the drug design to give new drug entities. Drug design strategies can be used to improve the compound's pharmacodynamic and pharmacokinetic properties. The resulting compounds from drug design go through series of preclinical studies and become clinical candidates if a compounds don't exhibit adverse effect or toxicity during in vitro and in vivo studies. So this is a process of an developing of an lead molecule. So we'll be looking step by step how it has been done. So first thing is that what is lead molecule? So lead molecule, it is nothing but a chemical entity and which serve as an moiety for an development of a new drug. And by inserting or by earlier also we have seen that by pruning method, so some moieties or some groups are being introduced or some groups are being cut, and after doing it, it has been optimized for an beneficial effects and minimizing its side effects. Many times it happens in case of a drug discovery that molecule is very potent and it is showing very good results, but they fail in this preclinical studies, and because they are showing in some side effects and these side effects are unwanted and which are not been desired, but due to heavy side effects the molecule are being cut down. And also there is a strategy to improve from a lead molecule that is pharmacodynamic and pharmacokinetic properties. So pharmacodynamic by inserting some groups, and also pharmacokinetic properties by inserting some groups, because earlier we have seen that some groups are responsible for this change in pharmacokinetic properties by increasing lipophilicity, hydrophilicity, and also in the earlier session we have also seen what kind of pharmacokinetic properties make a molecule more potent. And also we have seen pharmacokinetic steps, that is at absorption, distribution, metabolism and excretion. So all these four steps require different kind of an electronic parameters in a molecule. So earlier also we have seen in the earlier sessions. And the resulting compounds from a drug design go through a series of preclinical studies. So there is not only one drug which comes from a lead moiety. So there are thousands and lakhs of drugs which come from a lead moiety, and series of preclinical studies. Preclinical, it means that it has been done on animals, and once it has been suited in the animals and once the data it has been suited for an animals, then only the clinical studies, it means that on humans next study it has been done. And compounds which shows an adverse effect or toxicity, they are not being chosen as a drug candidate. So that may be in vitro or in vivo studies. Now we'll see the next point, that is strategies in search of new drug compounds. Firstly, it is an disease target. So once you know what kind of a disease it is, then we start according to it. Like nowadays, we are facing a big problem of an coronavirus, and once a pathophysiology it has been studied by an scientist, after that pathophysiology it has been studied by a scientist, and after that once we come to know an pathophysiology, so we will come across what receptors or what kind of an enzymes are responsible for that activity, and that has to be inhibited or that has to be cut down. After that drug target, so once we know what kind of activity it has been shown and what enzymes are responsible, from that we choose and drug targets. So there might be some endogenous ligands which binds. So according to that, one can find an antagonist depending on the structure of an agonist. After that, lead discovery. So lead discovery, it means that a compound which is responsible for an activity and it serves as a basic moiety for a drug design. After that, lead modification. So lead modification, it means that for designing and pharmacophore further. So a lead modification, it is done for an lot many purposes, like it should have an more active or more prone compound than earlier one. It should have an good pharmacokinetic properties also, which are responsible for ADME purpose. And also with that, by doing a lead modification also, it shows different pharmacodynamic properties. After that, preclinical and clinical development. So we have discussed a little bit earlier also that preclinical means it has been studied into an animals firstly, and once it has been studied into an animal, then only it comes to a clinical one. So clinical, it means that study it is done on human beings, and there are some phases in case of an drug discovery. So one of phase, that is preclinical studies, and after that clinical development. So basically, this clinical development, it has been studied on the humans, and data which is being analyzed mostly for an side effects of an drug. Already from preclinical studies, what potency of an compound it is or what potent it is, but in actual some of the data of an adverse effect, it is shown in the clinical studies, and many a times it happens that from clinical studies compounds are being cut down due to its adverse side effect. And also sometimes it has been happened that in post-marketing surveillance or clinical studies in the fourth phase, so there are total 4 phases, and in the fourth phase some of the compounds has shown a critical side effects due to which the compound has been banned. Once clinical development it has been done, and after that regulatory approvals for a compound or a new drug it is been taken, like patents it may be. Now we'll come towards an next point, that is possible source of lead compounds. So how to gain a lead compound and what are the possible sources of a lead compound. So first source, that is endogenous ligand, like example serotonin. So endogenous ligand, already serotonin it is present in the body, and based on that receptor, so serotonin it is responsible for a biological activity and which attached to a serotonin receptor, and from that, by making a modification with this serotonin, a lead molecule can be modified into an pharmacophore or a new drug compound. Secondly, by using natural products, so like ephedrine. So it has been obtained from an ephedra, and it serves as a lead molecule for a drug discovery. After that, animals. After that, microorganisms. Alexander Fleming discovered penicillin accidentally, and when he was working in the lab, so it was been shown that some microorganism of an like Penicillium notatum, which given an active metabolite, and that was nothing but an penicillin, and this penicillin it was responsible for an antibiotic activity, and it was been observed by Alexander Fleming, and then the discovery from an microorganism of a drug has been boosted, and there are a lot many drugs, mainly antibiotics, which are prepared from a microorganism. After that, chemical libraries such as combinatorial chemistries. So in the next session we will be coming across this, what is combinatorial chemistry. computational medicinal chemistry, that is drug design by using and computer models. So in case of this drug designing by using of by using this proteins and drug with different conformations, it is been interacted with this protein, and these proteins are been isolated. Protein means nothing but an ligands, and these ligands are been isolated from an organisms, and they are thoroughly been studied and what active site does they have. And this made computational chemistry or drug designing by computer aided drug designing. So this makes unable to an medicinal chemist, so without doing any type of an clinical or preclinical studies, medicinal chemist can directly interact a drug with an receptor, and once it interact with an receptor, the scores are, it has been measured, and depending on the score, a series of an compounds, it has been synthesized, and after that preclinical and clinical studies, it is been done in actual. So it helps an medicinal chemist to develop an compound rather synthesizing it firstly only to study with the use of a computer, only to bind to check and score of an interaction between proteins or between ligands and the molecules with different conformations of a molecule. So what should be the properties of lead? Firstly, pharmacodynamic property, that is it should have an efficacy, potency and selectivity towards a receptor. After that, it should have an physicochemical properties such as water solubility and chemical stability. So many of time it happens that compound it is not stable. So the property of lead molecule, that compound should be an stable one, and also with that it should have an water solubility. So it is responsible for an ionic interaction with an body fluids, and for that purpose we require an water solubility. After that, pharmacokinetic properties like metabolic stability, toxicological aspects. So these are important things. Once, like in some compounds, it happens that one compound it has been converted to an metabolite. So it is responsible for an either activity or either it shows and some toxicological effects. So these are important thing by of a lead molecule. Once it undergoes the process of an pharmacokinetic property, so it should have an metabolic stability. The metabolites which are been formed, they should be stable. And lot many times it happens in case of an anti-cancer drug, the metabolite which are been formed, so these are unstable or these are not good for a human one. And also toxicological studies, in case of some compounds they are very active, but due to toxicological effects they are been cut down or they are been removed. So toxicological aspects are also important for a lead molecule. Now we'll come towards a point that methods of lead identification. First one, that is random screening. Second one is non-random screening. Third one is high throughput screening. After that, structure-based drug design. So name itself it is indicating random. So it is showing an different activities and collecting lot many compounds and lot many activities they are been screened. Non-random activities, so they are little bit specified. After that, high throughput screening. So it is part of an combinatorial chemistry. Shortly it is said to be HTS. And after that, structure based drug design. Firstly, random or targeted screening method. So this method, it was being used before 1970. All the compounds, including synthetic chemical, natural products of a plant, marine and microbial origin from a given series are tested. In spite of budgetary and manpower overuse, this method may be used to discover drugs or leads that have unexpected activities. So by such random screening, antibiotics like streptomycin, tetracyclines were found out by these methods. So in these methods, some sources like synthetic chemicals, synthesizing lot many synthetic chemicals in a series, and after that natural products from a plant, and after that marine, and lastly from a microbial origin. So from this, all these sources, compounds are being isolated, and compounds which are being isolated, so they are being tested for different activities, and unexpectedly it comes across and that there are a lot many activities shown by a same type of a compound, and by this way streptomycin and tetracycline were the compounds which were found by this targeted screening method or random screening method. Now we'll come towards next point, that is non-random screening method. After 1970s, this method was invented. So earlier we have seen that random screening method, and it was before 1970s, and this non-random screening, it is after 1970s. So whatever the disadvantage of this random screening method was, so or limitations of random screening were, so this limitation has been overcome in non-random screening method. It is modified form of random screening which has developed because of budgetary and manpower restrictions, because when a compound, it has been sourced from many different sources like mineral source, animal source, plant source, or synthetic source, or some microorganism source, so it requires huge amount of an budget, and also it requires a huge amount of manpower to isolate the compounds, to test the compounds, and after testing to record the recordings of an testing of that compounds. And simply, in case of a non-random screening method, in this method only such compounds have a similar structural skeleton with that of lead are being tested. So similar structural skeleton, we will be coming across one of a term, that is bioisotopes, and so we will be knowing there what is this similar structural skeletons. So molecule has a similar type of an electronic parameters as well as steric parameters by using different skeletons, but similar in size and shape. And this non-random screening method, it is particularly only up to an lead molecule. So there are certain methods or certain modifications are being done on the lead molecule, and after that they have been tested. After that, high throughput screening, 10,000s of compounds against the target to identify any might be promising natural products, and synthetic compound libraries with millions of compounds are being screened using a test assay. So lot many compounds at one time, they are being introduced with an target, or they are being introduced for an activity check, and after that, once it shows some type of an activity, so specifically these compounds or these libraries or this series, they are being identified by separating the compounds, and once the separation it has been done, then every one compound it has been tested for an activity. So in this method, say suppose there are 10 HTS and only one HTS it is showing an activity. So all other 9 HTS they are been discarded and only one HTS which is showing an activity having lot many compounds in it synthesized from an combinatorial method. So they are been identified, and once they are been identified foreign structures and after they are been separated or they are been synthesized accordingly, and from that one compound it becomes an promising, or one compound it comes out and shows an activity. In theory, generating the entire chemical space of drug molecules and testing them would be an elegant approach to drug discovery. So checking each and every chemical rather than checking each and every chemical, so bunch of chemical it is been tested at once, and once we know that that particular bunch is responsible for activity, then only that compound it has been that compounds are been separated and they are been tested. After that, last point, that is structure based drug design. Three-dimensional structure of compound from virtual or physically existing libraries are docked into binding site of target proteins with known or predicted structures. Scoring function evaluates the steric and electrostatic complementary between compound and the target protein. Highest rank compounds are then suggested for biological testing. So scoring, so in case of this method, target proteins, they are available on internets or they are available with some agencies, and these target proteins they are been selected, and some structures without synthesizing by using an computational method or by using a computer, they are been their score it has been evaluated with a target protein, and once interaction of a structure with the target protein it shows a certain score, and this score it is used for testing of an biological activity. So highest rank compounds are suggested for an biological testing. Now so the first concept we seen, that is structure activity relationship. Shortly it is said to be SAR. The relationship between chemical structure of molecule and its biological activity. So by inserting some groups or by inserting some rings, so whether their biological activity changes, so that study it is said to be an SAR, and also stereochemical influences. After that, we have seen what are the factors considered for SAR, the size and shape of carbon skeleton. So in that we have seen insertion of an methyl group, so how the activity changes of an polyphenols with insertion of an methyl groups. At certain point there is restoration activity, but after that it loses the activity, or beyond it loses the activity, or below it loses the activity. After that we have seen the nature and degree of substitution. So what kind of groups they are responsible for lipophilic activity and hydrophilic activity. So we have seen that, and due to which ionic properties changes, and also the lipophilicity of a molecule changes. After that we have seen a stereochemistry of molecules. So we have seen certain structures, either the isomers are inactive, or either the isomers or enantiomers are active, or either they show a different effect. Like we have seen example of an ketamine, that R ketamine it is having an hallucinogenic effect, and S ketamine it is having a different effect. After that we have seen what is lead molecule, a new chemical entity that could potentially develop into a new drug by optimizing its beneficial effect and minimizing its side effect. So lead molecule it is and big process how that it has been developed. So we have seen that process like disease target, after that drug target it has been designed, after that lead molecule, or lead compound, it has been designed or been optimized from different sources. So these are the some sources to obtain a lead molecule, that is in endogenous ligands. So we have seen an example of an serotonin, after that natural products. So we have seen an example of an ephedrine, after that animals, example in microorganisms like penicillin, chemical libraries, like we have discussed little bit of a computational chemistry, and after that computational medicinal chemistry, that is drug designing by use of a computer. After that, we have seen properties of lead compound, that is, it should be pharmacodynamically, pharmacokinetically, and physiochemical properties, like with changes in little bit changes or modification in pharmacokinetic properties can be seen with changes of or modification in compound, also similar with an pharmacokinetic effect, and also with physicochemical properties. After that, we have seen methods of identification of lead compound, that is random screening. So it was used before 1970s, and it is used process for non-random screening. So it is specified to only a lead molecule. After that, HTS, high throughput screening. So it is like part of an computational chemistry. And after that, structure-based drug design. So it is based on virtual view only of interaction of a protein and drug. Thank you. We have completed module number 6, and this was lecture number 9.